So the final video in the course on protein synthesis, which is quite a big part of the course, and it kind of embeds itself throughout a whole bunch of different parts because proteins are so important, is what is this genetic code that we talk about? It's one thing to kind of throw around a word and go, yeah, it's all genes, it's all chromosomes, it's all proteins. Yeah, but if you don't understand what they're talking about, then we need to be able to break that down. So the genetic code is what we use to translate the mRNA into the language of amino acids. So it's not random. I don't just say AUU is, I'm just looking here, I don't just say it is phenylalanine just because I feel like it. And then next time it might be serine, and then next time it might be alanine in terms of amino acids. So it needs to be specific, and this is the code. You are always given this. You do not need to memorize the genetic code. If you have to answer a question that involves you translating the language of mRNA into the language of amino acids, then you will always be given the table. So please don't stress that you need to memorize the whole table. But what I am going to do is I'm going to show you how to read it. So what we do with this is we always look at the first position down here. So the first position are these bases here. So have a look if I have a U as my first base here. Then the second thing that we're going to do is we're going to go to the second position, and I'm going to say in this case it's going to be a G. Yeah, so these are the second position ones, and then the third one is going to be a C, and that's the third position here, and that's all of these letters. All right, so let's try and read it together and see how we go. So the first one here is I'm going to identify the U's. So the U's are all of these. Yeah. So anything in this whole one here starts with a U as its first base in the codon. Sorry. Then the next one are the G's. So the G's for the second position, which are in green, they now are going to cut us down to only this little block here. Okay, so this little block here is just all the ones that go UG. Now, we've got a few more options left. You could be UGU, which is going to be cysteine, but we want UGC. So here's the C within that bit. And so what we found out is that there's only one combination for UGC, and that becomes cis. Now, you don't need to know that that's cysteine. You don't need to know how to spell the full word. You can just use this 3-letter code when writing down your amino acids. And we're going to do a bit of practice on this in a second. So don't stress too much, but I'm just going to do one more example for you really quickly to show you what I want. So I'm going to say this one is CUA is my base. So I'm going to go to the C's in the first position. So it's going to be somewhere along this line. The second position is a U, so it's going to be somewhere in this block here. And the last position is an A, so it's going to be this one here. So when you put them all together, you can see that we have that combination there, and we end up with Lu, yeah, which is short for leucine. So when we talk about the genetic code, there are 3 main words that we need to use to describe it. So there are various questions that ask you about the features of the genetic code, or they might ask you about one of these words that I'm going to show you in a second. So the first thing about the genetic code is that it's always read in 3s. Yeah, always read it in 3 base pairs. And this has to do with the fact that there are 20 amino acids that are possible. So when you have a look at this table here on the right, you can see lots and lots of different combinations. Now, if I only had one base for each thing, and there are 4 combinations, I would do 4 to the power of 1. Yeah, and that means that we have 4 combinations. So if each base individually coded for an amino acid, I could have 4 possible amino acids, but I have 20 amino acids. Okay, so this is not going to work. So what we're going to do is we're going to go, all right, what if I had a combination of 2 base pairs? So when I go 4 to the power of 2, so that's 4 times 4 equals 16 combinations. This is still less than 20. So it's not going to work. So when we go 4 to the power of 3, which means, so this one would be e.g. just an A. This one would be e.g. a C. And then this one would be e.g. a C G. Yeah. When we go 4 to the power of 3, we end up with 64 combinations. That is greater than 20. So the 3 combinations here is the lowest number of bases that we can use to code for all the amino acids that we need to, because there are 20 of them. So we always say that the genetic code is read in 3s because this is the smallest combination of bases that can give us enough combinations to code for 20 amino acids. I'm happy to go over that with you if you need more help, but essentially you don't need to understand the maths. You don't need to replicate the maths that I've just shown you, but they're really helpful for understanding why it's read in 3s and not 1s and not 2s and not 4s and all of those different combinations. So it's always read in 3s. These are called codons. Yeah, so these codons here, oh, that's a bad color. These codons here are good. That's the name for the 3 bases on the mRNA. Just going to change that color one more time. But the anticodon is also in the tRNA. Yeah, but it's also in 3s. So it's always read in 3s. The second word we've got here is that we've got this word degenerate. Now, degeneracy means that sometimes you might also see it being called redundant. So either one of these words is, they're synonyms for each other in terms of this. And what it means when we say the genetic code is degenerate or redundant, it means that more than one codon codes for the same amino acid. Yeah, so the best example here is that e.g. UCU, UCC, UCA, and UCG all code for serine. Okay, and because they all code for the one amino acid, you can see that that's what we mean by degenerate. Why is that helpful? That's when we come back and we talk about mutations later on as to why it's helpful. But essentially, because of the fact that you could change this last base, you could change that U, you could change it to a C, an A, or a G, and it wouldn't change my amino acid. It would always code for serine. Yeah, and because of that, it means that some mutations might not have any effect on the overall structure of the protein, but that we will come back to later. The last word that we use when describing the genetic code is that it's universal. And what this means is it means that it's the same in every species. Okay, on planet Earth, we have the same genetic code. Doesn't matter if you're a monkey, if you're a virus, yeah. And this is why it's so exciting, this mRNA technology with the vaccines. I will come back and I'll probably do a special episode about the mRNA technology that we're using in the COVID vaccine. But it's why we can use human bases to create products that work against viruses as well, because viruses read the same genetic code. This is the language of all living things on Earth and non-living. Yeah, anything that contains a piece of nucleic acid, it's always read using this one code. That's pretty interesting that that has managed to evolve over time. Okay, so the last thing that I want to point out when we talk about the genetic code is that genes kind of have this particular part. They always know when to start and they always know when to stop. And what these are is these are called start and stop codons. So what you need to remember, you don't need to remember this exact combination, but I do want you to remember that methionine or MET is always the start codon, and I'll go back to that table to show you in a second. And then these 3 here will have stop written next to them, and they are always the final codon that we have in protein synthesis. What the reason is that their tRNA molecule, so if this is their tRNA molecule, I'm doing it just as my little clover leaf. Normally they have an amino acid on the back. A stop codon has no amino acid on its back. Yeah, which means that it can't keep contributing to the tail. I'm going to bookmark a little video for you to watch to show, to animate this process of translation, because often people can understand transcription, RNA processing, but it's translation that just makes itself a little bit trickier for your brain. So I'm going to link an animation to help you really understand what is happening during translation. So when I said you don't need to remember these particular base combinations, I'm just going to go back here and show you. You can see on this table here that we've got, that we have stop codons, which would be UAA and UAG and UGA. Now they're all our stop codons. And then in addition to that, we've also got one start codon. So every single protein in the human body or in any protein always starts with this one codon. It always starts with A, U, G. Yeah, that's how all proteins start, which is pretty cool to think that way. So what I would like you to do now is I'd like you to pause. And then I'd like you to go through and try and do the table on page 10 in your notes. So you're going to fill in the following. You're going to fill in the mRNA codons, the tRNA anticodons, and then the corresponding amino acids. And you need to use the tables that are in there. You've got the table that you get from VCAR, which is the one that I've been showing you, but there's also a little round kind of wheel like genetic code table. And if you find that one interesting, you can have a play with that. So we'll just have a look here and I'll show you how to do the first bit. And then we can see what we're talking about. So what we're looking at here is deciphering this genetic code. So you've got the DNA sequence is TAC. It undergoes transcription and RNA processing to become mRNA bases. Okay. Now, all we do there is we do the opposite. So if this one is T, the corresponding mRNA base is an A. So we're going to go down to this 3 bases here. The A, the corresponding base is going to be a U because we're talking about mRNA. So there's our mRNA bases. Now, what happens is we end up going through a process called translation and turning UUU into an amino acid sequence. All right, so I'm just going to look up mine. You might want to pause for a second and look up your table and see if you can find what is the amino acid that UUU corresponds to. So hopefully you've paused and you've come back into the video now, and you would have read your table to find UUU codes for the amino acid Phe or phenylalanine. Now, and this is the bare minimum that I want you to do in this table. I want you to make sure you've filled in the mRNA bases and the amino acids. Now, corresponding to this, there is also an anticodon. Now, it's rare that they've asked about it, but not impossible that they ask about them. So the anticodon is actually the complementary sequence to the mRNA codon. So you can see here in this first one over here, you've got A is complementary to U. Yeah, U is complementary to A and G is complementary to C. So when we're going back to figure out our tRNA bases in this second codon, we're going to go U is complementary to A. They're all complementary to A. Okay, so you should be able to work down this table now and figure out whether or not you can transcribe and then translate everything using the genetic code. I'll get you to pause one more time, finish it off, and then when you restart the video, I'll go on to the next slide, which will show you the answers. Hopefully you haven't cheated, and if you have, rewind a little bit so that you won't get the answers, because it is very, very worthwhile actually going through this task and making sure it's completed properly. So here are your answers. The quick way to check your answers is to look straight down your amino acid line here. So we've got phenylalanine, which was what I said. Phe was first. You might have written down ser, asp, glute, and through. Okay, so that's what you need to be able to calculate by the end of understanding the genetic code. If you can do transcription, RNA processing, translation, plus read the genetic code and understand the fact that it's read in threes, is universal and degenerate, then you're comfortable with protein synthesis. I'm going to produce one small little video that's going to talk through a schematic diagram, putting it all together. But other than that, this is the end of the protein synthesis series.